Pablo Camara, and it's my very great pleasure to introduce Dr. Vineet Bafna. So Dr. Bafna is a professor of computer science and engineering at UC San Diego, and a very well-known person in the field of bioinformatics for his many substantial contributions to a variety of algorithmic problems in genomics, in population genetics, and also in proteomics. Dr. Bafna received his PhD in computer science in 1994 under the supervision of Pavel Penn, who was at that time at Penn State. And during this PhD work, Dr. Bafna focused mostly on algorithms for multiple alignment and genomic rearrangements. And in particular, he introduced the breakpoint graph technique that is now widely used in the study of genomic rearrangements. So after this amazing work during his PhD, he moved to the Center for Discrete Mathematics and Theoretical Computer Science at Rutgers, where he stayed for a couple of years as a postdoc, and after which he decided to transition into the industry. He first worked at SmithKline, and then at Celera, where he became director of informatics research at the time of the Human Genome Project. A fantastic time to be there. And it's during this time that I believe that Dr. Bafna became interested in two other computational problems that he has continued pursuing during his career, namely the identification of peptides in mass spectrometry, and the problem of haplotype inference. In 2003, he returned to academia and joined UCSD, where he has continued doing fantastic work on a variety of algorithmic problems that mostly have to do with genomic sequences, such as haplotyping, as I was mentioning before, phylogenetic inference, genomic rearrangements, extrachromosomal DNA, and many others, as well as computational proteomics. He has been recently elected as a fellow of the International Society for Computational Biology for his outstanding contributions to the fields of computational biology and bioinformatics. And today we will hear about some of his most recent work on the role of extrachromosomal DNA in cancer. So without further ado, I let Dr. Bafna give his talk. Thank you so much, Vineet, for accepting our invitation.
Thank you, Pablo, and I'm really happy to be here. Let me share my screen and then maybe we can get started. So I should mention to everyone that when I was at SmithKline, I often used to come to Penn for their bioinformatics forum. It was more than 20 years ago or close to 20 years ago. So long time ago, but I have many happy memories of Penn from that time. So it's good to be back, even if it's virtual. Hopefully everybody can see my screen, and if not, just please feel free to speak up. I know that Babak had mentioned that we'll hold off on questions, but if there are short questions and you type them in the chat box or speak up, I'm happy to answer at any time. I don't have to finish everything that I set out to cover. Okay, I should also mention just a quick disclosure that I have a relationship with a company that is trying to commercialize some of the work that we are presenting here and developing therapy, which I'm not really going to talk about too much, but it's just wanted to disclose that conflict. So in a nutshell, my talk is really about amplifications of oncogenes in cancer. And these charts out here are examples of results that we often see today in the literature. This one is from Steve Ellich, and it's from 2017, but these things have been repeatedly noticed by people. And so if you look out here, these are all survival curves. It itself is a meta-analysis, so they take data from two other papers, and they look at the impact of mutation burden on survival rates and also the impact of somatic copy number amplifications, which they define loosely, and we won't go into their definition, but really refers to amplifications of certain regions. And so what they find is that individuals with high copy number amplifications have a much worse survival outcome than individuals with low copy number. And in fact, it's the inverse of the case when they have a high mutational burden. So if you have a high mutational burden, perhaps you're more likely to respond to immunotherapy, and you might have a better outcome at the end. So oncogene amplification clearly is a pathogenicity that must be considered in the context of cancer. And my talk mostly is about the rearrangements that cause these amplifications, right? So in a nutshell, what are the dominant mechanisms of somatic copy number amplification? That's for those reasons, I will not talk a lot about mutations in cancer, small nucleotide mutations, which really is the bulk of cancer genomics research today. So I should also have, I also have one slide for those of you who may not be familiar with how these analyses are done. But for most of you in the audience, this is probably something that you see on a day in and day out basis. But I thought just for completeness sake, it's useful to do this. So we are not at the stage where we can take a cancer genome and fully assemble it. In fact, we cannot assemble a genome of the complexity of humans, although now with the T2T consortium and other things, if you're willing to spend a lot of money, you might be able to do this. But instead, what we can do is we can take snippets of the donor cancer sample and map them back to the reference. And if you take, for example, in this paired end sequencing modality, if you take 2 ends of a 300 to 500 base pair fragment, when you map it back to the reference, you should have them mapped concordantly for the most part. But if you end up having the 2 fragments mapped very far apart, as we can see out here, then we can sort of conjecture that there's a deletion in the donor relative to the reference. And that causes this discordant mapping. Similarly, if you have a change in orientation, then we could, that refers to a rearrangement in the form of an inversion in the donor relative to the reference, right? So we will, as we go on in this talk, we will use these connections as a way of looking at rearrangements in the tumor genomes. Another sort of trick that people often use is just count the number of reads that fall within a certain region. So here, let's say that this is the redundant sequencing of the genome, and we expect every read to map, say, 10 times or 30 times, every base pair to be covered about 30 times or so. And if we see a complete absence of reads, then we can conjecture that there's a deletion. If we see more reads than we would expect to see, then that is consistent with an amplification in the genome, right? So these kinds of copy number changes can also be measured. They can be measured using arrays, but for the most part, we'll talk about sequencing-based approaches to measure these changes. And then the question again remains as to what causes copy number amplifications. Okay, so if you go open the textbook, you'll often see these kinds of duplication events, tandem duplications of large genomic regions, which could also be inverted tandem duplications or terminal tandem duplications. All of these different methodologies have been expressed, and for various reasons, these do not explain, or they cannot really explain, the high amplifications that we see in cancer. And this cartoon might explain why that is the case. So here we have the donor sample, and if it sort of replicates itself, then indeed, when you take a read from the junction and map it back, you will be able to get a signature which is indicative of a tandem duplication, and plus the copy number would increase. So that is good, but oftentimes we see copy numbers that are 10, 20, 30 times in a certain region. And the only way to explain them with the same sort of mark of discordant read would be to have the breakpoint be reused multiple times. And that, despite people looking at fragile regions and so on, doesn't seem to be so common. So if you had repeated breaks, it's unlikely that they would be exactly in the same region. And so for that reason, if you see very high copy number amplifications, these tandem duplications are not a good explanation. So we've been thinking about this for a long time, and we sort of went back to the to the original literature and some of this, and the model proposed by Barbara McClintock back in the 40s turned out to be something that people do begin to look at in cancer, have observed in cancer. It's called the breakage fusion bridge mechanism, and it's a very nice mechanism that can explain tremendous copy number amplification in a small number of cell divisions. And it starts with a break of a telomere, which destabilizes the chromosome. So the sister chromatids often fuse in order to stabilize the chromosome. But now this really creates the condition for another break, because the two centromeres must move apart during cell division. Right? So here's the cartoon, and when the centromeres move apart, you might end up with a break. And if you just sort of segment the genome in any way, and we won't worry about how it's segmented here, then this break is consistent with a region of the chromosome being inverted and duplicated, right? So we have this inversion and duplication, which of course is also increasing the copy number. Moreover, this end of the chromosome remains broken, and so the process can continue for a few cycles. And examples of these dicentric chromosomes are often seen in cancer, as well as other mechanisms, anaphase bridge formation, and these high amplifications in this form. So a few more cycles of this, and the genome is completely rearranged, as you can see. These regions, we are talking about very, very large regions. So some of these could be hundreds of kilobases long. Sometimes they can even be megabase pair long. And towards the end of this talk, we will show you some large regions which have a breakage fusion bridge operation, right? So you have here the tumor genome, but the tumor genome is not available to us in the form of sequence, but just in the form of short reads, right? So what we can do is sample from this donor and map it back to the reference, and we end up with these copy number counts, which are very high copy numbers for certain regions. So C here appears with copy number 6. You can just count it here. And A and B appear with copy number 4. Of course, in reality, these numbers are even larger. And so what we have is a copy number profile that we have obtained from the donor genome. And the question we ask is this explainable using a breakage fusion bridge mechanism? All right, so hopefully that question is clear to everyone. It turns out that this question is both combinatorially interesting, and it's just a very, very challenging problem. We worked on it for almost 3 years before we came up with a very talented postdoc in my lab by the name of Shai Zeka. He came up with the algorithm. And just to give some idea of the complexity, if you were to just take this A and reduce the copy number by 1, then in the pure form of the problem, this would be supportive of a breakage fusion bridge, and this would not be. And of course, one could say that this is close to copy numbers of a vector that supports BFP, but just in the pure form of the problem, the copy numbers on the right do not support a breakage fusion bridge operation. Right? So I won't give you the solution, but I want to walk you through some of the mechanisms that we can use to, some of the algorithmic tricks that we can use to get at these answers. So if you look at this slide out here at the bottom, we can think of this as the genome. The 3 segments are A, B, and C, and then we'll apply a few breakage fusion bridge cycles to it. And the only thing that we'll do differently this time is that whenever we have this prefix inversion and duplication, we will always write A on top of A, B on top of B, and so on. Right? So here's your first operation, which takes AB and does the prefix inversion and duplication. Here's a second one that takes AAB and does prefix inversion and duplication. Another one, and let's say we have one final one that takes the entire chromosome and duplicates it. And what we do as an algorithmic trick is we always take the count vectors and duplicate everything so that we have the symmetry that you see in the last slide here. But of course, the data is not given to us like this. The data is just given to us in the form of these copy numbers. So we have these copy numbers 12, 10, and 2, and we want to know if it's consistent with this kind of a breakage fusion bridge cycle. So if you look at this structure, you begin to see a hierarchy here, right? So you can take the 2 occurrences of C and sort of glue them together. And then you can also take the occurrences of B and glue them together, right? And then you can take A and glue those together. And you can see that there's this hierarchy where this looks like the root node of a tree, which has 5 children. And so the tree might look like this. This is corresponding to segment C, corresponding to segment B. And you can see that C has the 5 children given by these 5 pairs of Bs. And then each of these B nodes have in turn some or none A nodes as children, right? So we call these BFB trees. And one of the nice properties of BFB trees is that if you started from the root and did a kind of in-order traversal, you would end up with a breakage fusion bridge signal. Now, these trees are not just any trees, they have a lot of symmetries. In particular, you can rotate around any node and get the same tree back. And you can also rotate around any pair of nodes that are adjacent in the sense that they're the first occurrence of that node in this in-order traversal, and you will again get a symmetry about rotation. Moreover, they have long ends in the sense that the nodes towards the end, their distance from the leaf cannot be shorter than internal nodes. And when you have trees like this, then they are exactly breakage fusion bridge trees. And so one way to formulate the problem is that you take your copy numbers and you create trees that have these kinds of symmetries. And that's a very, very loose explanation. The actual algorithm is very, very difficult. And it's sort of almost impossible to explain in the context of a lecture. But like I mentioned before, Shai Zakov described this algorithm, and we published on it many years ago now. Right? And it gave us, for the first time, a tool to detect BFP, but it wasn't perfect because, of course, there are many variations due to noise in the data. And so we decided to both look at the noise and also incorporate some other signals. And this is one other signal that we incorporate. So in breakage fusion bridge, you often have a region of the chromosome that's inverted and duplicated. And if you were to sample from this chromosome and map back to the reference, you would end up with this foldback signature, right? So an excess of foldback reads is also a signature of BFP, and we combine this signature as well as the distance to a nearest BFP vector using Zakov's algorithm as a predictor for BFP, right? So how do we do? On simulations, of course, we do quite well. You can see that after we incorporate all of these signals, then in this case of simulated, where we take a genome, we simulate breakage fusion bridge operations and add noise, we can see with about 10% false positive rate, we have about over 80% true positive rate. So it's not perfect by any means, but it's a good tool to be able to detect breakage fusion bridge in genomes. And we are actually working on a revision of this almost 8-9 years after we originally did that work. So this was in 2012, the amount of genomic data was still somewhat limited. But when we started looking at the existing genomic data, we were somewhat disappointed. So first we did recapitulate a few examples of known BFP, and we also predicted one of our own in a pancreatic tumor sample, where the observed count vectors and the excess of foldback inversions made us predict that it was indeed a breakage fusion bridge cycle. But in general, we ended up with something that wasn't quite enough to explain the excess of somatic copy number amplifications that you see in cancer. Right? So in some sense, we had to go back to the drawing board. So just to summarize the discussion, tandem duplications are, do result in amplification, but are not sufficiently powerful to explain all these huge amplifications with very sharp boundaries. Breakage fusion bridge can explain those, but they don't seem to be as common, at least in our mining of the existing genomes. And then there are other mechanisms that have been proposed, and those explain complex rearrangements, but they are not typically models of amplification, or especially focal amplification of regions in cancer. So we went back to the literature again, and we started looking at these elements that were often called as double minutes. And this is, again, if you go back, you can go back all the way to Barbara McClintock's papers to see some evidence of genome circularization and so on. But in cancer, this was first reported back in 1965 in some neuroblastoma cases where people were looking at the cells in a metaphase spread. So in metaphase, the chromosomes are nicely compacted and they can be separated. And then when you do that in some cancers, you begin to see these what they used to call chromatin bodies, and later they call double minutes. On the right side, we have a figure from a paper by Geoff Wall. and they showed that these sort of double minutes are also evidence of DNA that's extra chromosomal in nature, right? So there was a lot of very nice biochemical work around this time in the late 80s and early 90s, but somehow this work fell by the wayside when the genomic revolution happened, because the genomic revolution allowed us to look at things at the nucleotide level, and it really increased the focus on single nucleotide changes, and people somehow lost context of the spatial presence, the spatial location of these amplification events. They kind of implicitly assumed that those were on the chromosome, perhaps in the native location, which we clearly see is not the case, right? So when we started looking at these around that time, Paul Michel, who's now my colleague on many of these experiments, came out with a very nice paper that I will talk about in a few slides from now. And that sort of got me in touch with him, and we planned this experiment where we started looking at many cells to see really how prevalent these double minutes are, or so-called. And we started calling them extra chromosomal DNA. And the reason is simply that people often used to look at these chromatin bodies in pairs, just to keep, separate them from debris. But we recognize that led to a severe undercounting. And the previous best estimates that people said were suggested, they used to suggest that they were found in 1.5% to 2% of all cancer samples. So we took a lot of cell lines and many images, metaphase images from those cell lines, close to 2,500 plus metaphase images. So we needed a computational tool that could look at all of these images automatically. And so this is a more recent incarnation of that tool. We initially just used plain computer vision methods where you look at a cell in metaphase that has been stained with DAPI. Here, just to illustrate the point, there's a FISH signal on a particular oncogene, which shows that you see the oncogene outside of the chromosome in an extra chromosomal setting, right? And the goal of this is just to take the DAPI signal, which is the blue stain dye that we see out here. Here we see it in grayscale, and to identify and count the number of extra chromosomal DNA particles. And so we originally, in the original paper, we used computer vision techniques. And more recently, we've switched to deep learning, like most others in the field. And we came up with this method that we call ECSEG, which takes an image like this and segments it pixel by pixel, and annotates the pixels as belonging to extra chromosomal DNA or chromosomes or intact nuclei. Right? And you can see that we do reasonably well in all of these classes, with perhaps chromosomes not doing so well, but in this intersection over union score, which takes the intersection of our prediction with the true prediction divided by the union. And you can see that we are very close to being perfect here. Here's another way of thinking about it. There's the recall, which is the fraction of true ecDNA that we get versus the fraction of predictions that are correct, which we call precision. And you can see that very nicely, even though there's a lot of variability in sample to sample, we seem to do a good job of prediction. We are comparing it here with the computer vision based method, where we focused mainly on precision so that we didn't have many false positives, but we suffered in terms of recall. And so we were really grossly undercounting the DNA. But most of the results from that paper I will show you are still based on the old data. We have since gone and done the data, but that's unpublished, right? And so the tool at least works to some extent to be able to predict and count the number of ecDNA per cell in a sample. Okay, and so in 2017, when we published this, the results were quite shocking to us, at least. When you look at the data, let's look at panel C here, you can see that in normal, we see ecDNA very rarely, but in cell lines and even in immortalized cells, and certainly in patient-derived xenografts, we see a huge proportion of samples. On the average, about 40% of the samples that we looked at showed ecDNA. And we see that across many different tumor types, even though the prevalence does vary across tumor, you can see that there's a huge proliferation of ecDNA in samples all across different cancers. And so this sort of suggests that ecDNA could be a dominant mode of oncogene amplification in cancer. And our work from that point onwards has really been about elucidating the fine structure of ecDNA and sort of building out its functional role in mediating cancer. Okay. All right, so let's talk about an ecDNA model of amplification in cancer. And I'll explain the model in using a cartoon, and not everything is sort of well proven. We are still working on many aspects of it. And of course, not us, just us in general. Many researchers are working towards many topics, and many questions remain unanswered. So, so the model really starts with the random breakage and formation of a circular extrachromosomal element. Okay. So fragments break off at random and form extrachromosomal elements, right? As you know, on human genome, there are many origins of replication. And so the replication and initiation and recombination can lead to multiple copies. Even if we assume that ecDNA is replicating at the same rate as human chromosomes, we still would have as many copies, at least in the beginning, right? Now, the fun thing about ecDNA is that they do not have centromeres, right? So they don't have centromeres, which means that when the cell divides, they will segregate independently. And it's quite possible that if you have two ecDNA, both of them land up in one daughter cell, and the other daughter cell is free of ecDNA. Right? And so this process can continue, creating tremendous heterogeneity, cell-to-cell heterogeneity of ecDNA, but by itself, it would not cause a change in copy number. All right, so the other sort of tenet of this model is that the ecDNA are not as tightly bound to chromatin as chromosomal DNA would be. And so genes that are present on ecDNA could be highly dysregulated and could be overexpressed. And so if that is the case, and if you're really unlucky and if you have an oncogene on the, or some kind of a proliferative gene on the ecDNA, then cells that carry more copies of the ecDNA are likely to be more fit in this context, or more likely to grow and divide than cells that do not carry ecDNA, right? So in the next generation from here, you might end up with more cell divisions from this lineage compared to this lineage, and that results in an increase in copy number. And if you sort of do that modeling, then in a few cycles, you might lead to both tremendous heterogeneity, but also tremendous amplification of the oncogene. Okay. And then finally, and perhaps somewhat controversially, these ecDNA might reintegrate back into the chromosomes and show up as homogeneously staining regions, where there are large copies of ecDNA that have accumulated in one region. And at that point, they are very stable, but they are grossly amplified. And if you were to take these bulk cells and do whole genome sequencing and look at these paired end signatures, you will see both complex rearrangements, but also tremendous amplification of certain regions without necessarily the breakpoints being reused. So it's a very clean model that explains copy number amplification without having to have the same portion break off at exactly the same boundaries over and over again. All right, so it's an attractive model, and it seems that many of the tenets of this model do appear to be true. And so I see a question, I'll quickly take it, and then in the remainder of the talk, I will go through and talk about some of the aspects of this model. All right, let's take a question, if there is, but except that, what's the minimum size that our DL system can detect? So that's a great question, John. Our method cannot, it can only detect things that are really visible under the light microscope. Most ecDNA are quite large. They are 1 million base pairs, and those we can detect quite easily. There are examples of small circular elements in the chromosomes. Sometimes, circular elements that are like hundreds of base pairs or thousands of base pairs. those are not large enough to carry genes. And so we actually don't call them ecDNA. In the literature, they are sometimes referred to as ECC DNA, extrachromosomal circular DNA, and that's an unfortunate sort of conflict of nomenclature, but we are really talking about a different event. And then there's a quick other question, which is the incorporation in the chromosome well established or common? And Yusuf, I will answer this question by and by. We are not the only ones who say this. Okay, so let's go ahead and see what we can say here. I'm sorry, I'm going in the wrong direction. All right, so first let's just settle the case that these are indeed circular elements. And so here's on the right, you can see an electron microscope image of these elements, and you can very nicely see both the circular structures, but also a replication of ecDNA forming these sort of double minute like structures. Okay. All right, so the question that Yusuf asked, ecDNA might integrate back into chromosomal regions and explain many focal amplifications. So I mentioned earlier on that, with my colleague Paul Michel, before we started, we had the chance to read a very nice paper of his, and it's probably time to talk about that a little bit. So they were looking at a glioblastoma cell line. This is a brain cancer, and it's marked by amplification of EGFR, which is an oncogene that's commonly amplified in glioblastomas. And you can see on the left side that when you look at the naive cells in metaphase, EGFR appears as to be on ecDNA in great numbers. So when they treat this cell line with the drug erlotinib, which targets EGFR, mysteriously all of the ecDNA disappear, right? And instead you see an HSR-like structure, a chromosomal structure with a gross amplification of EGFR. And then when you remove the drug, these things come back together again, right? So it was a very interesting experiment, and a lot of people scratched their heads as to what could be happening. And although they gave some clues here, people often ask, is this a population effect? Like there could be that both of these cells are in the population, but when you give the drug, this cell, this population dominates, and then when you remove the drug, the other population comes back, and they really represent different modes of amplification of EGFR. So we started looking at this now with genome sequencing data, and we developed this tool called Amplicon Architect. And the goal of this tool was to really get at the fine structure of ecDNA, and we use what you might call directed assembly method. So rather than de novo assembling things, we map everything back to a reference and use the reference as a template to reconstruct the architecture. And so here's sort of a quick overview of how that algorithm works. It's only for specific regions. So we start with a seed interval that you can see is amplified. And then we gather discordant reads that are very tightly linked with this amplified regions, right? So this suggests that this region out here gets connected with this region. We also do a copy number analysis, and wherever we see a sharp break, even if there wasn't any discordant read there, this is suggestive of a rearrangement at this location. So at that point, we do a much more sensitive search for breakpoints for discordant reads that emanate from these breakpoints. And then we combine all of this data. We do a bit of smoothing using a flow algorithm, and that sort of gives us an amplicon graph, a graph where the segments, the nodes correspond to genomic segments and edges correspond to connectivities between these segments. And then we can extract paths and cycles from this graph. And the cycles, could be representative, we'll talk about that more later, could be representative of extrachromosomal DNA structure. But for now, we'll just look at some of these graphs, right? So in this paper that we published a few years ago, we did this experiment with the same model system that was in the original Nathanson paper. So here we have the glioblastoma cell line, and you can see that in many cells, many replicates, these are all bulk cells, when we reconstruct using Amplicon Architect, we get an identical structure. The same ecDNA comes out saying that, all these cells have a common ecDNA, an ecDNA with a common origin. And then when you apply the drug and you again sequence and assemble, you actually get the identical structure back. It doesn't look like that here in this low resolution, but when you look at it, the structure is identical. You see all of the elements of the cycle, except that it's much lower in copy number. Okay, so the copy number changed from nearly 200 out here to something like 10 out here. But moreover, you see the emergence of a new extra chromosomal DNA, right? And this one contains the oncogene MDM2. So it looks like a mechanism of resistance that nevertheless keeps the tumor proliferating. And then when we remove the drug, an identical structure comes back out again. And if these were indeed different populations, you wouldn't see the same structure being maintained. It suggests that the same element actually goes inside the ecDNA and comes back out again. And this is not a perfect answer to Yusuf's question, but we have a lot more evidence. Since then, even in this paper, we show some more, but now, and I'll talk a little bit about some long reads and so on, but I won't show all the data here, but we do have a lot of evidence that this indeed happens. Moreover, if you see out here, not only does this ecDNA come back again, it is linked to the MDM2 containing ecDNA, and subsequent FISH expression, FISH-based validation shows that these ecDNA co-localize, suggesting that the two ecDNA recombine into a more complex form. So a lot is happening out here, which tells us about the dynamics of ecDNA, and some of that is subject of ongoing research as well. All right, so moving on, we talked about, we mentioned that, dysregulation and the fact that they might not be as tightly bound to chromatin could cause overexpression of oncogenes on ecDNA. And so we worked on this on some cancer-derived cell lines, including GBM39, in a paper that also we published a couple of years ago. And it was straight sort of a functional omics experiment where we applied a whole bunch of techniques, including RNA-seq, ATAC-seq, and also some high-chip and other chromatin capture-like methods. And the main advantage that we had here was that because of our tools like Amplicon Architect and so on, we had a very good idea of the segments that belong to ecDNA, and we could contrast those with the same segments in other cells where they didn't form ecDNA, as well as other segments on the rest of the genome, and those form the control of our experiment. So you can already see that if this is the normal copy number of WGS out here, this is a sort of a key, then this suggests that there's an overexpression of ecDNA. And on this cell, you can see that here's the normal, and the WGS is uniformly expressed to something that is 100-fold more. So there's a tremendous amplification of the gene. Similarly, when you look at the genes that are located on these ecDNA, hopefully that also addresses a little bit more of John's concern. So we have intact genes in there, including EGFR, and we can see that these genes are also 100-fold more expressed. The ATAC-seq signal also shows tremendous accessibility of genes that lie on ecDNA. And then finally, when we look at these chromatin region interactions, you can see that in this U87 cell line, which doesn't carry the ecDNA, so it's like a control. If you look at these distant points, they do not interact as one might expect, because they are presumably far enough apart, more than a couple of million base pairs apart, that they would not be in the same sort of topological domain. But when you look at them in GBM39, where they are part of an ecDNA, you see all these strong interactions, suggesting that they are spatially close in this rearranged structure. So all of this paper, of course, I'm doing, I'm doing sort of disservice to this paper, which was largely written by my collaborators, where they do a lot of different functional studies to show that genes that are expressed on ecDNA are likely to behave very, very differently from genes that are expressed on the chromosome. All right, so that, all of this work you could do on cell lines, where we can do the metaphase experiments and confirm that we are finding ecDNA. But when we have patient data, we often don't have live cells, and that's a big challenge. We can extract DNA and we can do sort of whole genome sequencing, and you run amplicon architect to get at cyclical structures, but we don't quite know where ecDNA are. And so this was some of the things that we started thinking about. And we did something that's very naive in retrospect. And we are still, I mean, this amplicon classification is still something that we haven't published quite, and we are still working on it. But in this paper, what we did was we just took all the amplifications and just gave them different categories. So specifically, if we could find large regions, more than 10 kb, that were well amplified and formed nice cycles, we call those circular amplicons. And then there were others, which I won't talk about too much, but there were some that showed heavy rearrangement, but did not show the completion of a cycle. And then there were others that were, not very heavily rearrangement, but showed some rearrangement and some amplification. And so we did all these classifications and suggested that the ones that we call circular are most likely to be ecDNA. And then we tested this back on the cell lines for which we had metaphase data. So here's sort of our predictions in a nutshell. So on the y-axis are the outcome of the imaging metaphase experiments, where we count the number of ecDNA per cell, right? So it goes all the way from 0 to a very, very large number. It's on a logarithmic scale. And clearly, if you have large numbers of ecDNA, then that cell is ecDNA carrying. So this, if you draw a horizontal line somewhere, everything above that line would be ecDNA positive. Everything below that line, you could call ecDNA negative. And then on the right side, we have the whole genome-based analysis, where we, on the x-axis, where we call something as being circular if they can complete cycles. And this is our prediction of an ecDNA, right? So how do we do? Well, let's say that we draw a cutoff here, which says that if you see an ecDNA once every 2 cells, then we, I don't know, it's not conservative, but it's a way of saying that this cell carries ecDNA. And then we say that everything that is circular is ecDNA, right? So in a more computer science or computational terminology, everything out here on the top right quadrant is true positives, things that we predicted and showed a high number of ecDNA per cell. Everything in the bottom left is a true negative. And then out here in top left, we have what we might call false negatives, things that had ecDNA, but we could not predict. And bottom right is what we call false positives, right? So false negatives, we sort of gave up on, because there could be many reasons why we could not complete a cycle. And in the absence of long reads, it's hard to predict. So we don't worry too much about false negatives, but we do want to get rid of false positives as much as possible. And so we looked at a few examples. Okay, this is a very nice example because we also had some experimental data here. So on the left is the same erlotinib treated GBM39. I'm sorry, on the right side. And we know that when we give the drug, the copy number reduces, and then we remove the drug, it comes back up again, giving a dynamics to the ecDNA going in and out of chromosomes. But on the left side, we have another cell line. This one is HCC827. It's a lung cancer cell line, where when you treat it with erlotinib, it carries EGFR amplification, but erlotinib has no impact on the copy number, right? So when you look at this data in the metaphase, it becomes very clear. On the left side, the HCC827 really shows up as homogeneously staining regions, but it also has some peculiar recurring pattern, which suggests that this could be a breakage fusion bridge. On the right side, of course, we know that it's ecDNA. Right? And when we looked at the copy number profile and we applied Shire's tool, we could see that this was very, very strongly suggestive of BFP, but we wanted more direct evidence, right? So it was time really to delve into some long read technology to try and deconvolve the structures. Okay, so why long reads? I think for people who are in genomics, I think this is not a difficult slide to understand, but maybe this picture is still important for people who may not have seen this kind of data. So imagine that you have this complicated amplicon graph where you see 3 copies of this segment A and 1 copy each of the segment B, C, and D, right? And so this is consistent if you just did short read sequencing, you would see that the head of A is connected to the tail of C, the head of C is connected to the tail of A, and likewise with A, B, and AD. Okay, so all of these adjacencies can be easily identified with a short read structure. But then when you try to reconstruct, you will get two equivalent structures that support all of these breakpoints. And you can see that they are different because here you have C followed by D followed by B. Here you have C followed by B followed by D, right? So you have these two alternative structures that are completely consistent with the short reads. And these kinds of ambiguities make it difficult to reconstruct uniquely with short reads. But if you had a long read that spanned this region, right, then you could break the symmetry and say that B followed by D is the correct answer, and B followed by C followed by D is probably not correct. So this single long read allows us to disambiguate and say that this structure is the correct one. Now, here's the challenge though. We are not talking about normal genome assembly where these repetitive regions are tens of thousands of base pairs, but rather we are talking about extra chromosomal structures where some of these repeated segments are hundreds of thousands of base pairs. And so we really need extra long reads, right? So we started looking at the longest possible reads that we could get, and we settled on these optical maps. And for those of you who haven't seen it, the optical map technology doesn't give you sequence, but it actually gives you these little markers of restriction enzyme sites. And then when you compare your optical map against sort of a sequence where you can in silico mark the positions of these labels, then the distance, the conserved distance between the labels suggests that these two come from the same region and allow you to do the mappings. And I'm actually, I'm going to run out of time, so I'm going to go a little bit quickly. We use these ideas to develop a tool that can disambiguate these structures, and we call this tool amplicon reconstructor. And so when we apply it to HCC 827, and I'll sort of skip some of the details on this, we see these very nice inversions and duplications, and we end up with a 9 million base pair structure, perhaps the largest breakage fusion bridge that completely explains a set of breakage fusion bridge cycles that create this amplicon. So we do have a handle with these long read technologies of other mechanisms of amplification. And now we are going back and reanalyzing some of these data sets if we can. Okay. All right, so we also applied it to some other cases. I'll skip this slide in the interest of time. Sorry, my screen froze for a minute. Sorry about that. All right, so we also applied it to some other cases where we had false positives, and they really looked like extra chromosomal DNA, except that we couldn't complete the circle. And this is because these extra chromosomal DNA had reintegrated back into the chromosome. And you can see on this picture out here that we see this region being duplicated and inverted, and somewhere here as the points of integration. And it's all sort of suggestive, though not a proof, of this agglomerative model that we are currently working on, where multiple ecDNA can recombine, form larger ecDNA, and these then integrate into a single genomic location, forming what we call homogeneously staining regions. Okay, so it's not complete data, but just to say that after doing that and after separating out some of the cells as being breakage fusion bridge derived, we do get a reduction in some of our false positives. And now just using sequence data alone, we have a pretty good handle on what ecDNA we might see. Okay, so we applied this in a paper that we published last year to almost all of TCGA, everywhere where we had whole genome sequence available, so about 2,000 plus samples. And we also took another data set from the PCOG project from ICGC, and we combined all of that data, and you can see the results are not identical, but they match many of the results that we had earlier seen in cancer-derived cell lines, in the sense that ecDNA are very, very common all through different tumor types, although they appear in different prevalences in different tumor types. And with the brain cancers again being dominant and the solid tumors being a little bit more than some of the leukemias. And these extra chromosomal DNA are never seen in the matched normal or in the blood tissue derived from these individuals. Okay, so we can start doing these functional analyses as we showed in this paper, and where we showed that not only is higher copy number explained, but when you have ecDNA genes, then the expression is even more than what you might just explain using increased copy number. Okay. So when I started this talk, I mentioned that people carrying somatic cell amplifications have indeed poor outcomes, and we can now revisit this data in the context of ecDNA. And indeed, we can show that individuals that carry amplifications have worse survival outcomes. But even among those individuals, when you subset those two individuals carrying ecDNA, they have even worse survival outcomes, suggesting that ecDNA are indeed the source of a lot of amplification and many pathologies. Okay, so I was asked to keep the time of the talk to within 45 minutes. I'm a little bit over, and I just want to conclude by just reminding everybody that ecDNA are these large circular structures, typically without a chromosome, and the model of independent segregation and selection really allows for rapid modulation of copy number. I only mentioned increasing copy number, but the same mechanism also allows for decrease of copy number. And then we have some results that I didn't show, which is that a lot, or in fact, a majority of oncogene amplification in cancer can be explained via ecDNA formation. And then, because of the gross functional differences between ecDNA and chromosomal DNA, we see a lot of functional changes on genes that are on ecDNA. And so because of this, the importance of this, there's a lot of work going on in ecDNA, just understanding the biogenesis and maintenance of ecDNA, and many other labs whose work I haven't been able to cite in my talk have actually been working on this. Okay, so I see a question, but this is really my last slide. So let me just acknowledge people, and then I'll be happy to take the question. So many people help me, of course. The collaboration with the Michel Lab has been tremendous. Paul Michel just recently moved to Stanford, but we still work very, very closely together. And specifically, Kristen Turner and Sean Wu in his lab, I presented much of their data in my talks today. And then we also collaborated on the pan cancer project with Roel Verhaat, and Hoon Kim in his lab did a lot of that analysis. In my own lab, Shai Zakov and Marcus Kinsella had worked on breakage fusion bridge many years ago. Utkrish Rajkumar and Doruk Bader worked on the image analysis pipelines. And then Viraj and Jens worked on these Amplicon architect and reconstructor-based tools. And Nam Nguyen, who is a postdoc, helped with a lot of the analysis. So I'd like to acknowledge all of them. And I'd like to thank you all for listening. Thank you very much.
Thank you so much, Vineet, for such a fantastic talk. We have time for questions. I see Babak is one. I don't know if you want to unmute and ask it, or you want me to read it.
Probably if you read it's easier, that would be more organized. But
okay, so so in one of your slides you showed a significant difference between the frequency of extrachromosomal DNA in solid versus blood cancers. Is there any reason for such differences? Also, would you comment on whether there is any association among the extrachromosomal DNA frequency and dynamic with therapy mode, for instance, DNA damaging agents versus cell signaling inhibitor?
I mean, these are all great questions, and unfortunately, I don't have good answers to any of them, especially for the solid tumors, why they're more likely to show ecDNA. We actually don't know. And we don't know that this is indeed true. This is just an observation that we had in the specific cohort that we looked at, the TCGA, but I think we have seen other examples where we do see extrachromosomal DNA in some leukemias too. So the jury is still out on that question. Now, with respect to therapy, again, there's not as much data because people haven't quite associated therapy with ecDNA. We do know that ecDNA, the copy number modulation is much more rapid than the accumulation of mutations. And so these cancers arise so quickly and grow so quickly that they don't have time to acquire a lot of excess somatic mutations. And so that is one of the reasons that they are probably less amenable to many of these immunotherapies or therapies that might target neoantigens or specific mutations. So that is definitely the case. There have also been many examples now, or at least like 5 or 6 examples where people are trying to target amplifications, and the cells seem to have a lot of ways to escape that. They can do that either, as in the example that I showed, by having the ecDNA escape into a chromosome, or they can do it by increasing the number of copies. So, just increasing the number of ecDNA and overcoming the impact of the drug, or by generating ecDNA which are completely different, which were not presented. We have seen all such examples not in patient data, but in treatments of cell lines with cancer. So I think there are many mechanisms that unfortunately a cell has to escape. And if you look at targeted therapies and so on, they will always have the danger that if they are targeting a tumor with ecDNA, that the tumor will find some way to escape. And perhaps the right strategy would be to target the mechanisms of ecDNA formation and or proteins that are involved in ecDNA maintenance, rather than targeting the oncogenes that appear on ecDNA.
A question along the lines of what Joseph was asking you in regard with the incorporation back into the chromosome. When you look at the insertion points of when it incorporates back, is it completely random or preferred places? And also, is it known the mechanism in terms of, is it more like a non-homologous enjoining or homologous recombination, the way of inserting back? Like how does it work? Can you get some insight basically by looking at the insertion points?
Also a great question. So we do not have enough examples to conclusively say one way or the other, but all the evidence that we have suggests that it's a random insertion point, that there is not, there aren't sites that are particularly susceptible to integration. They are often in different chromosomes, and but we do see an agglomeration of ecDNA prior to insertion. So it isn't, if you have 100 copies of insertion, they do not insert in 100 locations. And there is a paper that's on bioRxiv from our collaborator, Howard Chang, where they provide a physical basis for this. They suggest sort of a hub structure where ecDNA are not freely floating around, but are actually agglomerated on hubs. And that might provide a framework. It's a sort of protein-based scaffold, but it provides a framework where ecDNA could recombine and form larger structures, which would then integrate into one point. And we do, and I think the fact that people have cytogenetically observed many HSRs, which suggest a lot of amplification in one region. All of our results with HSR suggest that these are the non-native location for the gene. And so ecDNA is the simplest explanation, that ecDNA agglomerated and entered that. Now, with respect to your question about non-homologous end joining versus sort of repeat mediated, it's interesting because when you study the breakpoints, which is how we do it, why everybody does it, then it is almost always a non-homologous end joining mechanism. It's very rarely the case of non-allelic homologous recombination, and we are not the only ones. Many other researchers have pointed this out. However, some unpublished data to share with you is that in 1 or 2 examples where we looked at, and now that we have these long reads, we can really look at it very nicely. We find that a region of, the region where it integrates, there's a small duplicate that is created of that region. And so even though the breakpoint itself looks like non-homologous, there's actually, you call that region A, and that region A, the way the HSR works is that the A binds to the extra chromosomal DNA, and that comes back into a copy of A and then reintegrates. And so So the actual mechanism might be more complicated. We don't know what that might be. Okay, and then there's some other very wild theories. There's a paper from Royal Verhaeghe group where they suggest that these ecDNA are actually bouncing around and they act as mobile enhancers. So they come in contact with chromosomal regions and change the expression of the genes where they come in contact with, but also that contact could allow for recombination and integration to happen at that location. So a lot of questions that are not quite known.
And I have another question, just out of curiosity, it's kind of a little bit tangential. So there is the work of Vilera, Holmes, and Bachmann defining those spaces of phylogenetic trees, or spaces of trees in general, and they have very nice mathematical structures and so on. Have you or any other group look at how BFB trees place in those spaces? I guess they should define some subspace out of those spaces, but have people studied those? Do they have any mathematical structure that is interesting?
It's also a very nice question. To the best of our knowledge, nobody has studied it because I don't think it's easy to collect that kind of data. We would probably, the best way would be to have some single cell data and to look at BFB structures. But when you look at bulk cells, if you look at breakage fusion bridge structures, they won't be so different. Like you see heterogeneity, but it's not easily mapped onto a tree structure. And we are working on a paper where we have some time course data, but it's very, very limited. Like we have 2 or 3 time course samples, and we see a progressive change in ecDNA structure, but it's not enough to say that there's, like we don't give tree structures, there's some progression and progressive change in ecDNA structure is the only thing that we can surmise from there.
Very good. So if there are no other questions, well, thank you so much again, Vineet, for such a fantastic talk and your meetings today. I think you have still at least one more meeting.
Thank you, Pablo, and thank you all for inviting me. It's a pleasure to speak. And Babak, maybe we could go online right away, if you don't mind afterwards. Okay. Bye, everyone.
Sure. I think I need to change the link.
Zoom. Yes, absolutely.
Thank you.
All right. See you, everybody. Thank you. Bye-bye.
Bye. Thank you.